Discovering a promising new molecule in a laboratory is a significant achievement, but moving that discovery into the public domain involves immense complexity. Distributing a potent chemical to millions of people requires a rigorous transition from a controlled lab bench to the unpredictable reality of human biology. Rushing this development path leads to severe consequences. Flawed safety data can mask toxicity. Human trial subjects can be subjected to unethical conditions, and manufacturing errors can result in contaminated or ineffective medicine. National and international regulatory gatekeepers, including the FDA, enforce specific frameworks to prevent these outcomes, acting as a mandatory filter for any new treatment. Pharmaceutical compliance is built on three sequential phases: good laboratory practice, good clinical practice, and good manufacturing practice. A drug must successfully pass through each before it can legally move to the next. These stages provide the verifiable evidence required to prove a drug is safe and effective. Failing to follow this sequence leads to regulatory rejection and creates avoidable risks to human life. The process begins with good laboratory practice, or GLP. This framework applies strictly to non-clinical studies conducted before any human testing is authorized. GLP ensures that foundational safety data is reliable. Researchers use this phase to conduct toxicity studies and evaluate a drug's pharmacological effects on animal models or cell cultures to determine if the product is safe enough for initial human exposure. As a managerial quality control system, GLP dictates how these studies are planned, recorded, and archived. This ensures that every result is traceable and that the conditions of the experiment can be verified by outside auditors. By standardizing the organizational process, GLP ensures the integrity of the data used to justify clinical trials. It provides the documented proof that a molecule has met the safety requirements necessary to begin testing in humans. When a drug moves from the lab to human volunteers, the regulatory focus shifts to good clinical practice, or GCP. GCP is rooted in the ethical principles of the Declaration of Helsinki and the Nuremberg Code. It establishes the international standard for protecting the rights, safety, and well-being of trial subjects, making informed consent a mandatory prerequisite for participation. Beyond ethics, GCP ensures that trial results are credible. Professional monitors and biostatisticians oversee the data collection process to confirm that the drug's efficacy is measured accurately and without bias. These rules are codified in the ICH E6 Good Clinical Practice Guidelines. This harmonized standard allows clinical data generated in one country to be accepted by regulatory authorities worldwide, reducing costs and speeding up the delivery of new drugs. GCP provides the scientific and ethical evidence that a treatment is effective in humans, while ensuring that participants are treated with the highest standard of care. After a drug is approved, it moves into mass production under Good Manufacturing Practice, or GMP. This is the final phase of the cycle. Manufacturing requires a massive shift in scale. A process that worked for a few milligrams in an R&D lab must be adapted to produce kilograms of the drug while maintaining absolute chemical consistency across every batch. GMP compliance relies on unannounced inspections of facilities and meticulous control of the production environment. Manufacturers are required to report any quality deviations or accidents to the FDA to ensure the public is protected from manufacturing errors. This system ensures that every pill produced in a factory is identical in purity and potency to the ones proven safe during clinical trials, eliminating the risk of batch contamination or variability. We can view these three phases as a geographic map of drug development. GLP governs the laboratory, GCP governs the clinic, and GMP governs the factory. These systems connect through a unified quality management system. This flowchart shows the cycle of planning, evaluation, and improvement linking them. Results feed forward. Lab safety data informs clinical trial design, which sets factory quality standards. This creates a continuous chain of documentation and verification. If any single link fails, the drug cannot be legally sold, as the integrity of the entire development process is compromised. These regulations ensure that the journey from raw scientific potential to a finished medicine is supported by verified data and ethical practice. For more deep dives into the frameworks of modern biotechnology, Subscribe to